Okay, in this video, I want to go over some of the basic methods used in cognitive neuroscience. That's the specialty at the intersection of cognitive psychology and neuroscience, where we investigate how neural circuits affect and control and basically underlie cognitive processes. Now, plenty of cognitive psychology is done without a direct focus on the brain, without looking at things through a neural lens or worrying about that lower level of how it all happens. But when we do look at that level and bring in that focus for cognitive questions, we're doing cognitive neuroscience. So the goals of cognitive neuroscience, they include things like identifying functional areas or networks in the brain. For example, what areas are active when you speak, or what areas are necessary to imagine, or how do areas in the occipital lobe connect to areas of the temporal lobe in a way that lets us visually recognize a zebra versus a car. We're also trying to identify how neural processes are involved in cognitive activities. For example, how are memories stored at the neural level? Like, how is a memory stored in the brain? Or what neurotransmitters are involved in learning a new skill? Or how do electrochemical action potentials coming in on axons from our retina in our eye, how do those get turned into the perception of seeing a square? Likewise, you might be interested in understanding individual differences, like why some people have worse auditory working memory than others, or higher IQ, or whatever, or gender differences in, say, spatial processing, or how maturation and development of the brain maybe affects maturation of cognitive processes and abilities, or things like neuroplasticity, flexibility, and change in the brain. And some cognitive neuroscientists may be interested in how to assess or treat brain damage, or how to develop brain-machine interfaces that might, allow a paralyzed person to control a robotic prosthetic arm, or may someday be a standard implant used by everyone, like an internal smartphone, but direct to your brain. At any rate, that's just a sampling of the kinds of things cognitive neuroscientists are aiming to do by scientifically studying the role of the brain in our cognition. But now let's see some of the ways this research is done. So first off, we can study living humans who happen to have suffered some brain damage. This is the area called neuropsychology. For example, we might compare different patients who have brain damage to different areas and are showing kind of different cognitive problems. Like we might have one person, patient X, who suffered a stroke to one part of the visual centers of his brain. And now he's suddenly unable to recognize and identify objects or give their names. But he does fine with other cognitive functions, like he's just fine with the spatial aspects of vision. He can recognize what's close or far or moving using vision. He just can't recognize and name and identify objects, right? Meanwhile, over here we got patient Y, who also had a stroke, but to a different part of the visual centers in his brain, and now he's unable to use spatial information coming in through sight, but he does fine recognizing and identifying objects and giving their names visually. So they've got this kind of opposite issues, right, with a couple of basic visual functions. So if patient X has damage to a specific place, like the lower pathway out of the occipital lobe, and patient Y has damage to a different part of the upper pathway out of the occipital lobe, then we have some evidence that those functions are specific to those areas. We start to kind of map out how the brain lets us do things like visual perception or categorizing objects. Or alternatively, a neuropsychologist might study patients with brain damage just to understand how recovery happens, like how much the brain can adjust after injury through neuroplasticity, but also to study and learn what treatments might best help the recovery of cognitive function after a part of the brain is damaged. Regardless, though, these are generally case studies because we're usually looking in depth at individual cases. So it's not like an experiment where we might take 100 people and randomly assign 50 of them to get brain damage. Thus, neuropsychology, it can help us form theories and hypotheses and do some basic testing of theories, especially if we also run sets of these patients through some experiments. But we're always limited by the fact that nature did the sorting into the brain damage group and the control group. It did the sorting for us. And we're also limited by the fact that an injury to the brain might not cleanly damage just a single location. Like a stroke might damage one large chunk of the occipital lobe, but not all of it, but also maybe a bit of the bordering parietal lobe or maybe some other bordering nearby area. So it's not as clean as we might do if we were doing an experiment from scratch, right? But it lets us at least see the I don't know, the basic, I don't know, we'll say the develop hypotheses about the basic structure of things. So let's go to this idea of dissociations, the basic logic of a lot of neuropsychology studies for learning about brain functions to develop these basic hypotheses. So imagine you are Pierre-Paul Broca, a physician in France back in the 1800s, and you notice, you're a doctor, right? You notice a number of your patients, they have trouble speaking after a stroke. And this stroke is to a specific little area in their frontal lobe. Now, always, if that little area is damaged, the person has trouble getting words out and using grammar rules to form even simple sentences. They're having trouble getting language out, producing it, speaking. They also, have trouble, saying the difference between like the boy hit the girl and the girl hit the boy. However, they can still understand language that they hear. Like, they know what you're saying when you say boy or when you say girl or car or giraffe or whatever. So they understand language when receiving it, but they can't get it out themselves. They can't produce it. It makes sense. If you're Broca, it makes sense that you'd hypothesize that this area you keep seeing damage, that might be the speech center for your brain, right? For producing language. What we've got here is a type of evidence we'll call a single dissociation. Damage to a specific area impairs one specific function, A, but not some other function, B, that might be closely related. So another example might be a patient who can identify friends. So someone comes in, you're a neuropsychologist, they come in, they're having these symptoms. They find that they can identify their friends and family members by voice, right? If they're hearing them over the phone or just hearing the person shout from another room, they have no problem telling who that is. They recognize, they can name that person, but not by sight. They can't recognize their face. If the person just stands there or they see a picture, sight doesn't allow recognition. In that case, we might hypothesize that the damaged area, the part of this person's brain that was damaged, might be related to visual face identification and assume that some other part of the brain does voice-based or auditory identification. That makes sense, right? But that would be a single dissociation, that type of evidence. Meanwhile, a stronger form of evidence in neuropsychology comes from what we call a double dissociation. This is where we have sets of patients with damage to two different areas of the brain where their functional impairments are kind of exact opposites of each other. Like, damage to one area impairs function A only, but function B is fine, while people who have damage in a different area, that impairs function B only, but function A is just fine. That's a double dissociation, and it gives much stronger evidence that those areas really are involved in function A and B, respectively. So, for example, if we've got patients with damage to that area that Broca's discovered, and they all have impaired language production and speaking problems, but they can understand language fine, that's a single dissociation. But we have a double dissociation if we also keep finding other patients who have damage to this other part of the brain, maybe a little area back in the temporal lobe, where those patients all have an impairment of language understanding. They have trouble comprehending words, but maybe they can produce language and speak fluently just fine using gibberish and wrong words that they don't understand. That would be a double dissociation. Visually, you can kind of think of it this way. A single dissociation, like on the left here, gives us evidence like this graph. So our patients are the red line. They're doing worse on task 1, but just fine on task 2. While our controls are the blue bar, right, or the blue line, and they do fine on both tasks. That doesn't necessarily tell us that a cognitive function is localized to that one part of the brain. At most, it just tells us that these are two different functions, but maybe our patients are doing worse on task 1 just because it's the harder of the two tasks or something like that. Meanwhile, on the right side, the right graph here, you get the idea behind a double dissociation, where we can compare two different sets or different types of patients. So the red group, they suck at task 1, but they're fine at task 2, and vice versa for the blue group. That, assuming they, each group has a consistent specific place in the brain that's damaged for each group, that gives at least decent evidence of some independence or modularity of function in the brain. Although this is still a little limited. Generally, this method falls apart for more complex neural networks, things involving lots of interconnected areas. Now, a real life example of double dissociation in the research literature would be situations like patient RM who has perfectly normal visual perception after some brain damage, totally normal visual perception, can see things just fine, but lost the ability to use visual imagery, like can't imagine visual things, no mind's eye. Meanwhile, patient CK might have damage to a different area, and suddenly CK has issues with visual perception, like with just seeing things, but visual imagery wasn't affected at all. The mind's eye still worked like normal for CK. This tells us that visual perception and visual imagery are distinct things, even if they recruit a lot of the same areas in common. And it tells us there's at least one area that is specific to imagery processes only, but not involved in perception, and another area that's specific to perception processes only, but not involved in imagery. Or other examples you'll see might be patients with damage to one part of the hippocampus suddenly might not be able to remember, drawings that they've seen previously. Hippocampus, remember, that's kind of associated with making new memories. So we do these tasks and we're showing them some drawings. If they had damage to the hippocampus, these people can't remember the drawings they've seen even just 10 minutes ago. But they do still have one ability intact. They can copy a drawing that's in front of them just fine. But meanwhile, we might have a different set of patients with damage to this one specific part of the parietal lobe. And these patients have the opposite problem. Again, consistently, the same damaged area, they consistently have the same opposite problem. Now with these people, they can remember drawings they've seen previously. Things we showed them 10 minutes ago, they can say, yeah, I've seen that one before, or no, that wasn't one of the ones I saw 10 minutes ago. But they can't copy one that's right in front of them. So that helps us distinguish these two different areas and might have two very different functions. A good analogy for understanding double dissociations is the TV analogy. So if your TV, let's say, loses the color, but the video is still coming in as black and white, so you're still seeing the image, but the color's gone. If that happens, you could conclude that general video transmission and color information, they must be distinct processes, right? They're distinct processes, but that's a single dissociation. They may not be fully independent processes because you can't lose the video and still have the color, right? But let's say instead you have two separate TV sets that are damaged. Two separate TVs. One where the sound is broken, but the video works just fine. And a different TV where the video is gone. There's no video, but the sound is there. In that case, you can conclude that video and sound are two independent functions in a television. That's a double dissociation. At any rate, neuropsychology is studying people with brain damage. It's only one type of cognitive neuroscience method. Like I've said here, it's method one. So another method would be studying someone's brain after they're dead. Like, imagine your patient has a certain condition or disorder, but maybe we don't know why, or we just need more details that we can't get while they're still alive. Maybe for your patient, there was no traumatic brain injury or stroke or anything that shows up in a CT scan or an MRI. Maybe it's something they've had since birth. We may not be able to answer the questions, but after death, a post-mortem exam, an autopsy basically, could help us identify some underlying problems in their brain or differences in their brain. For example, a definitive diagnosis of Alzheimer's disease actually requires examination of brain tissue, and doing so can help us understand the underlying causes and the possible treatments by investigating things like plaques and tangles and other issues in certain brain tissue. So in other situations, we may be able to get some useful info from the brain by scanning it while they're still alive, yeah, but we may gain a lot of additional info in a post-mortem. So this has also been used in professional athletes, like football players with chronic traumatic encephalopathy, where basically repeated head trauma, like repeated concussions or getting knocked out a lot if you're a boxer or something, those add up to major brain issues, and we can answer a lot of questions about that doing a post-mortem exam. Also, autopsy of a brain after death, it's also been useful for studying some rare conditions, like the most famous case study in cognitive psychology, maybe in psychology in general. It's this guy named H.M., who after a surgery at age 27, he lost the ability to form new memories. Now, when he died in 2008, after decades of studying him as a case study and doing like single subject experiments with him, scientists actually live streamed the post-mortem dissection of his brain. And you can even look up the 2,400 individual slices of brain tissue online. And later, scientists doing papers analyzed that tissue. It led to a lot of new debate and more nuanced understanding of the brain areas that he lost in that surgery. So sometimes post-mortem exams are used to answer these sort of cognitive neuroscience questions. Meanwhile, other cognitive neuroscience overlaps a little more with behavioral neuroscience by using animal models. So one of the biggest benefits of animal models is that it allows us to do single cell recording, meaning single neuron recording. Basically, we can attach an electrode to individual neurons and see when they fire, which helps us identify what that neuron is tuned to, what makes that neuron go off, what its function is. So you might see a study like this one from 2015, where they measured the activity of individual neurons in the brain of monkeys while they played a multiplayer video game. Basically, it's a controlled laboratory way of studying cooperation, so we can then start to get at the neural basis of how cooperation works or social function in primate brains like ours. Now, as the abstract says here, you probably can't read this, but I'll read it for you. As the abstract says, a cornerstone of successful social interchange is the ability to anticipate each other's intentions and actions. While generating these internal predictions is essential for constructive social behavior, their single neuronal basis and causal underpinnings are unknown. In other words, we don't know how neurons in the brain manage to predict social partners' behavior. Here, they say, we discover specific neurons in the primate dorsal anterior cingulate that selectively predict an opponent's yet unknown decision to invest in their common good or to defect, to betray. And also, they identify specific neurons that encode the monkey's own current decision, just based on the prior outcomes, working with this partner. So basically, in this study, they recorded from individual neurons, and they found individual neurons that seemed to encode a prediction of what their partner would do in a prisoner's dilemma sort of game, basically predicting if their partner would betray them or cooperate in this game. And also they found individual neurons that seemed to predict what they themselves were going to choose soon afterward. So that's the kind of study that'd be hard to do with humans, because recording from individual neurons like that requires surgery into the brain in order to implant the electrode that records each neuron. So it's a benefit of animal studies. Another technique with animal models is lesioning. So creating a lesion, which means selectively damaging or removing part of the brain to see how it affects cognitive processes. Like after we shut down area X or area Y, how does it affect their rate of learning or their memory performance or their emotional reactions or whatever? This lets us have experimental control and precision in a way we can't get when studying humans who have brain damage by accident, right? Where, as I mentioned, in humans, the damage might be diffuse, it might be partially overlapping different areas. We get a lot more control here in animal studies. Now, obviously this sucks for the poor animals involved. So like all animal research, there are some major ethical questions, and this kind of thing definitely requires very heavy oversight by an institutional ethics board that includes veterinarians and community members, in addition to other scientists to make sure the science is done right. But thankfully, we do have some techniques now to do lesion studies on animals without permanent damage. What we do is just kind of temporarily shut down an area of the brain. Like one method is to cool it, to get it really cold so the neurons can't fire for a while until it's brought back up to normal temperature. Now, there are other animal model techniques becoming more and more common in cognitive neuroscience today, like gene editing studies that involve raising animals that lack certain cells or certain receptors, or that have cells or receptors they wouldn't otherwise have as that species, just to kind of see how that affects their behavior and their cognitive processes. So for example, the chemical substance PTC. It's a chemical substance that for us humans, it tastes super bitter. People will taste it and be like, that's really bitter, right? To humans. But mice don't taste it. They get nothing. It's totally neutral. It's not bitter to mice. They don't detect PTC in that way. Yet, with gene editing, we can give them our human gene that expresses that bitterness detecting receptor, the gene that, when it activates, creates the proteins that forms these receptors on the tongue, the bitterness detecting receptor, and suddenly those mice are able to perceive something that they normally couldn't. They can taste something they normally can't taste. That's an example of gene editing, and sometimes that's used in cognitive neuroscience studies too. We won't go into too much detail yet. Meanwhile, though, most cognitive neuroscience done by cognitive psychologists doesn't use animal models, nor does it use people with brain damage, but rather uses brain imaging. In other words, brain scans to measure a perfectly healthy living human brain in action. Now, a big benefit here is that this allows for at least some kinds of genuinely experimental research designs where we manipulate the cognitive activity that the person is performing, that's our independent variable, and then we observe which brain regions become more active or less active as a result. That's our dependent variable. This can help us identify what brain areas are important for what cognitive functions and test a lot of hypotheses that would be harder to test in neuropsychology patients or animals. And it also gets around some of the worries about whether we can generalize conclusions from people with brain damage or from species that aren't humans. Now, one of the most common ways of brain imaging is to measure electrical or magnetic activity. Electricity and magnetism are tightly linked, so measuring one or either of those that kind of happens along with neural firing. So remember, neurons, they have an electrochemical process. There's electrical activity happening when a neuron fires an action potential. So basically, we're measuring that electrical activity. That would be methods like doing an EEG or an MEG. I'll explain what those are in a minute. But one thing to note about methods that measure electrical and magnetic activity is that this tends to have really good temporal resolution, meaning we get accurate info about the timing of when neurons fired, but it may be more limited in spatial resolution, meaning it's not as good for localizing exactly where in the brain the activity happened. Alternatively, some methods of brain imaging actually measure blood flow in the brain. Basically, when one area of your brain activates above baseline, like it changes how much firing it's doing compared to usual, like when you weigh the future risks or benefits of two choices, part of your frontal lobe has more activity than normal. That activity of neurons, it burns through glucose and oxygen. And neurons themselves, they don't store glucose or oxygen inside. So we need blood to transport that stuff to basically kind of give them refills, basically. So we get extra oxygenated blood going to those areas after the neurons fire there, right? After a bunch of neurons there fire a lot. And lots of deoxygenated blood then leaving the area. So big brain scanners like PET scans, PET, or fMRI scans, they take advantage of this fact with clever ways of measuring blood flow, which then lets us infer what areas of the brain were just active, just before this. Now, these types of brain scans, they can be really good for localizing things, meaning they have really great spatial resolution. We know precisely kind of what cubic millimeter of brain was just active, but they're bad for timing information, meaning their temporal resolution is at least a little poor, since it can take multiple seconds before the change in blood flow registers in the part of the brain that was recently extra active. Now, in a second here, I'll explain each of the techniques above in more detail. But also, there are other techniques I want to at least mention briefly, like TMS, where we can actually activate or shut down an area of the brain temporarily, similar to those animal lesioning studies where we cool things off, but in this case, safe and non-invasive from humans. We don't have to go into the brain at all to do this with humans. But let's start by looking at the imaging, the brain scanning techniques above. Let's start with the ones that measure electrical and magnetic activity. So to start with, there's EEG, short for electroencephalogram, which literally just translates to electrical brain writing. Basically, we put a cap on their scalp that has electrodes to measure electrical activity out there at the scalp, so this isn't inside the brain. And this works okay because when the neurons underneath there in the brain are firing, some of the electrical impulses basically spread around and bounce their way up toward the surface in tiny amounts that we can pick up based on a lot of neurons all going off in the same area at the same time. So we measure this electrical activity in response to some event or change, like sometimes we're manipulating to test a cognitive hypothesis, right? We might measure which areas of the brain have activity at what time point when someone's doing a memory task on a computer, or when a baby is listening to their mom's voice versus a stranger's voice, or when looking at a normal face versus an upside down face, or whatever it is we want to test, right? Again, it captures timing very precisely because electrical activity happens fast. but it's measured at the scalp, so it's not very spatially precise. The signal is sort of diffused and bounced around quite a bit by the time it gets through the skull from the area where the neural firing actually happened down there in the brain. But with enough electrodes, like 100 or more electrodes spread across the scalp carefully in a research setting, we can actually deduce location somewhat decently. Now, because the electrical signals are so noisy, usually we'll actually measure over hundreds of trials. Like if we're measuring math problems with an EEG, we might have them do hundreds of the same kind of math problems, hundreds of trials, and then we average it out to find the time course at which parts of the brain were active when. So that averaged out response, that's called an ERP, an event-related potential. If you ever see a cognitive neuroscience study reporting ERPs, that means they're just using an averaged EEG readings in this kind of way to answer their cognitive questions. So you might see cognitive psych studies like this, where they used high-resolution EEG to study dynamic brain activity during video game playing. Now, don't worry about the details here, but basically they were interested in timing information and synchronized waves of activity, brain waves, in different areas, like the frontal versus the parietal lobes, and EEG was able to give that kind of information. Meanwhile, there's MEG, short for magnetoencephalography, which just translates to magnetic brain writing. Basically measuring magnetic fields outside the head using a scanner like you see here in the picture. The reason it makes sense to measure magnetic activity is that electricity and magnetism are tightly linked. That's why you hear terms like the electromagnetic spectrum for light waves, right? So action potentials in neurons, they're doing their electrical thing when neurons fire, that's also having effects on magnetic fields in that area. And like EEG, this means it's accurate for timing, but maybe less accurate spatially for exactly where the activity happened. Meanwhile, like I said, there are methods that instead measure blood flow. So for example, PET scans are one type, where PET is short for positron emission tomography. Here we inject just a harmless radioactive tracer into the bloodstream, which then goes wherever metabolic processes are happening. Like that would include blood flow going to the brain. So then a detector measures radioactivity, kind of like a specialized Geiger counter three-dimensionally around the head. A detector measures radioactivity coming from different brain areas. So the area with the highest radioactivity after a task tells us that that brain area used the most oxygen during the task, which tells us that area had a lot of neurons firing and thus needing the blood flow to replenish them after. So if we inject the tracer and then have someone do a bunch of math problems, it's a cognitive task, right? Do a bunch of math problems, we can figure out what areas of the brain are involved during mathematical problem solving. We could also compare one type of subject to another using this method, like comparing male brains to female brains during math or during language or during perceptual cognitive tasks, or comparing those with a disorder like schizophrenia or depression to control participants. Now, waiting on blood flow means it's not very precise for timing of neural activity, but it is precise spatially. Location of activity, we can get pretty damn good here. And since you're probably curious about the idea of putting radioactivity into a volunteer's body for scientific study, the average PET scan might involve around 14 millisieverts as the dose of radiation. Just for comparison, that's roughly the same as spending 1 year living in Denver. Denver is the mile-high city, which is just a little closer to the sun's rays than a city at sea level, and that makes a difference of about 14 millisieverts just by living there for a year, okay? And a PET scan, that's about the same, around 14 millisieverts. So it's definitely more than a dental x-ray or a chest x-ray, but those are already way less than the amount of normal natural radiation we're all exposed to every year, which averages around 2 millisieverts. And pilots, like people who fly in airplanes, so they're up a little closer to the sun for at least some of their workday, they average around 3 millisieverts per year from flying, and more if they take certain international routes that get them closer to the sun. And a CT scan, by the way, that you might do in a hospital, that's around 7 to 10 millisieverts, pretty comparable to a PET scan. So you wouldn't want to do a bunch of these studies in the same 5-year period, but one time is not much risk as long as there's full informed consent about it. That said, nowadays a newer technology is much more popular for brain scans, which is to use an MRI machine, the same one you might see in a hospital for doing an MRI on your broken knee or something like that. Use an MRI machine, but in a way that's specially programmed for fMRI, which means functional magnetic resonance imaging. So here we're using basically giant magnets inside the machine, but they're measuring bits of iron in oxygenated or deoxygenated blood that's flowing into or out of parts of the brain after neural activity. So again, we're deducing oxygen consumption from that blood flow, and from that oxygen consumption, we can deduce where neural activity just happened recently. We do this during cognitive tasks, and then we can identify what areas of the brain are active for task 1 versus task 2, right? For math versus language, or music versus language, or math versus computer coding, right? Or some cognitive task versus a control condition of just resting, or perhaps resting versus meditating, right? All the while, we're seeing what areas of the brain are firing more than usual or less than usual, which this is an important thing to note. Anytime you see these pictures of a brain scan with certain areas colored in, that color was added where neural activity went up or down, but often we're talking a change from baseline of just a couple of % of neural firing. In other words, that area was already active, just like our whole freaking brain is pretty much always active and has a lot of baseline firing of neurons all the time. So what we're seeing in these diagrams is areas that have, say, 1% or 2% or 3% more firing than during rest or during whatever our comparison activity was. If we scanned someone doing word tasks in their native language and then doing word tasks in their second language, the results might be displayed as one of these lit up brain images, but the lit up parts might just be the parts that differed between those two tasks, even though a bunch of other areas in the brain were equally active during both tasks, but don't get colored in on the picture. So when you see brain scan pictures, you're not seeing what areas are active. You're seeing differences in activity, changes in activity. It's a subtractive method. So you're seeing comparisons, and often those differences are very small. Likewise, the colored in area might be reflecting areas where neural firing went down, went lower than baseline or lower than during the comparison task. So don't be too confused by colloquial language about an area of the brain lighting up or some area firing when you smell your partner's scent or look at a scary picture. The brain, one, it isn't literally lighting up inside, right? It's just neurons firing. And the picture that you see, the displays like this that have different parts of the brain colored in, that just tells you where some changes in processing were happening, even if it's just to turn down one area slightly or turn up one area slightly. Now, just like PET scans, this is like a blood flow technology, a blood flow measure. So it can be super precise spatially if you use the right settings on an fMRI, but timing info isn't as ideal, especially since it involves a delay of up to 6 seconds or so while you wait on blood flow changes. Now, depending on the settings you use, you can get better temporal timing, but it's just never quite as good as measuring electricity directly. So here's a quick summary of these kind of 4 of the most common brain scan methods. A couple detect neural firing fairly directly through electrical or magnetic activity, and their temporal resolution is good. Well, the others detect some things more indirectly through blood flow, but we can get some really good spatial resolution. Now, I mentioned there are other methods, including one I wanted to mention here called TMS, though this isn't technically brain imaging, but rather brain manipulation. So TMS stands for transcranial magnetic stimulation. Transcranial meaning outside the cranium, outside the skull, and then magnetic stimulation. So with TMS, we're basically applying a magnetic field from this little wand instrument we use to turn up or turn down parts of the brain, to activate or shut down a specific part of the brain temporarily. Now, the big benefit of a technique like this is we can safely create temporary brain damage wherever we want and measure how it affects cognition. So we can create our own double dissociations without causing permanent harm. This lets us do some really cool experimental designs that we can't do with just brain scanning. Kind of think of it this way. With brain scans, we used cognitive function as the independent variable. We control what task they're doing, like math or language or whatever. And the dependent variable we're measuring is brain activity. But with TMS, we can do the opposite. Control their brain activity, turning certain places on or off as an independent variable, then measure how that affects cognitive function as a dependent variable. When I shut down this area back in the occipital lobe, the person temporarily goes blind, although their hearing and touch and other senses are fine, as is all their thinking. When I turn up this area here in the parietal lobe, I turn it up, more firing, spatial attention gets affected, right? Or when I zap this area up in the frontal cortex, maybe it affects their risk-taking or their planning or their ability to imagine a contrafactual where things in the past could have gone differently. And by combining both of these types of experimental designs, doing it both ways, right, opposite types of designs, we can really start to test and rule out hypotheses about how the brain underlies cognition of all kinds. Now, there is a limitation with TMS, and that's that it can only affect brain areas pretty close to the skull, meaning the stuff somewhat near the surface of the brain. Thankfully, that does include the cerebral cortex, those 4 lobes we've talked about where lots of cool stuff happens. So we can do a lot with TMS, but we can't, say, shut down the amygdala when we want to test hypotheses about the role of emotions in financial decision-making or something. That's just too deep into the brain. So here's an example of what a cognitive neuroscience study using TMS might look like. In the title here, SMA means supplementary motor area. This is just an area right up next to M1 in the frontal lobe, where we talked about like voluntary motor signals originating before they send out commands to the muscles. So this is an area near there. They're just using TMS on an area just in front of the supplementary motor area. So disrupting pre-SMA activity, so area just to the front of the supplementary motor area, disrupting activity there impairs facial happiness recognition. So they say, if you read the abstract, they say, it has been suggested that left pre-supplementary motor area could be implicated in facial emotion expression and recognition, especially for laughter and happiness. To test this hypothesis, in a single-blind randomized study, we investigated the impact of TMS on performance of 18 healthy participants during a facial emotion recognition task. So skim over the details here, but they delivered TMS pulses to either the pre-SMA area that they're testing or to a control location elsewhere on the skull, just called the vertex. It's like a body part of the top middle of your skull. Sure enough, disrupting the pre-SMA area made people worse at facial recognition, like facial happiness recognition, worse at being able to tell when someone was laughing or happy, though it didn't affect fear or anger recognition, nor their reaction times. So basically, past research using brain scans has shown this area was active when people did an emotion recognition task, especially for happy faces. And now in this additional study, they've shut down that area and people can no longer recognize happy faces. Together, that kind of stuff gives us pretty strong evidence that this area is an important contributor to processing positive emotion on people's faces. Now, there are other cognitive neuroscience techniques that we won't get into here, like diffusion tensor imaging, which is useful for measuring which white matter areas in the brain connect to which other areas, kind of get a feel for the organization and connections. Or there's near-infrared spectroscopy, which uses electromagnetic radiation, just similar to light waves, but a little lower energy than red light, more towards the kind of light energy picked up by night vision goggles or a TV remote control or AM radio waves, that kind of thing. Or using optogenetics, where we might do a genetic modification, like making certain cells deep inside the brain become light sensitive, like our eye cells are. And then we can activate those by shining a form of laser light that can travel through the body, which then activates those light sensitive cells deep in the brain. But the details of those methods, stuff like that, that's a little beyond our level. Now, we can occasionally do single neuron recording. Similar to what we saw with some animal studies, we can occasionally do single neuron recording or single neuron stimulation in live human brains if they're already going to be undergoing some brain surgery and if they decide to sign off on volunteering and helping science along the way. So this is surprisingly easy to do for cognitive studies because there are no pain receptors inside the brain. So actually, brain surgery is already often done with the patient awake the entire time. We already do this just to check in with the patient to make sure during the surgery we're in the exact correct location. Like poke the occipital lobe and they'll mention seeing a streak of light in their visual field. But if they volunteer, we might also test some cognitive neuroscience hypothesis while the brain is open, but before the surgery itself is carried out. So for example, we might hook up an electrode to a single neuron in the auditory cortex and measure how much neuron Y fires when this person hears a high tone or a low tone or a pure tone or a complex tone or a voice or whatever. Then we can measure neuron X and neuron W and other neurons nearby. Or if they're a musician, we might test some hypothesis about, measuring like what area of the brain has been implicated in musical processing. We might test that stuff. Or we might have them do a language task where we measure an individual neuron in some part of the language network. That's kind of the idea. In fact, this method was made famous by studies like those of Wilder Penfield back in 1937, for example. When doing some surgery for epilepsy, he did some tests like this. And he observed and he questioned awake patients while stimulating neurons in a location in the brain kind of near where the parietal and the frontal lobes meet. Basically what we saw before called S1, the primary somatosensory cortex, it's this strip in yellow here, and M1, the primary motor cortex, that's in red here. So he'd hook up an electrode, just hook it up to a random neuron there in one of those regions, say in the yellow strip. Then he'd stimulate that neuron X we'll call it, and the person, he would ask the person, or he would observe what happens. Like they might say, I feel a tingle in my left big toe when you turn that neuron on. Then he'd stimulate the neuron next to it, right? Go to the neuron right next to it. From neuron X goes to neuron Y, turns that on, and they might say, I feel a tingle in my left second toe, right? The one next to the toe they felt before. And he did this over and over and over again for different neurons all over the place, mapping out what the person felt when he turned on a given neuron in the yellow strip. In that somatosensory cortex, S1 that we talked about. Then he'd put the electrode on a random neuron in the red strip, what we call now the motor cortex, right? So then he moves the electrode to some random neuron a little further forward in the back of the frontal lobe there, and just see what happens when he turns that neuron on. So he uses the electrode, zaps it with a little electricity, makes the neuron fire. Sure enough, if he stimulated neuron U, their right index finger would twitch. They didn't choose to do it, but they just made a movement because some neuron in their brain was activated by his electrode. While if you move next door to neuron W, might make their right middle finger twitch. And not far away, stimulating neurons A and B might make their left index and left middle fingers twitch, and so on. And the neurons that controlled movement of a given finger, they were all next to each other in the brain. While the neurons that controlled movements of the next finger over, they were in a nearby clump. But the clumps of neurons that controlled the toes were way further away in the brain. Although the clump for one toe was next to the clump for another toe. And those were all near the clump for moving foot muscles and ankle movement and so on. So by the time he had kind of systematically mapped this out, tried a whole bunch of neurons in different places and seen where they were and what they did, that strip at the back of the frontal lobe that made some muscle in the person activate, or that strip at the front of the parietal lobe that made a person feel a touch on some part of their body, he mapped all that out and he realized the layout was really organized. Like the neurons for touches to your elbow are next to the neurons. In your brain, they're next to the neurons for touches to your forearm, which are next to the ones for your hand. Likewise, when it comes to the motor side of things, the movement side, right, the frontal lobe little strip there, the neurons controlling your trunk and your hip movement, they're next to the neurons controlling knee movement, which are then next to the neurons controlling foot movement, and those are next to the ones controlling toe movement, and so on. So what his work did, all the way back in the early 1900s, was demonstrate that the brain has a logical map of the body, both for touch input and for motor output. And of course, this same method could be used not just for the touch area or the motor area of the brain, could also be used for any other area of the brain. So if we activate a neuron in the occipital lobe at the back of the brain, they might see a little dot or a flash of light. He also showed that certain areas have more neurons, basically more cortical space devoted to them, so that the body map inside our brain isn't exactly to scale. Basically, we need precise motor control of our hands to manipulate objects and our lips to talk and manipulate food. So we've got a lot of neurons in the brain dedicated to controlling those parts of the body. Likewise, for sensing things, just for survival reasons, we get a lot of sensory feedback from our hands and fingers, right? And also from our lips and tongue for like food and things like that. So we've got a lot of neurons dedicated to processing input from those parts of the body. But we call this exaggerated representation of brain neurons. We call it the homunculus. It just means little man. It's like the little body or map of a body inside the brain. The body-shaped map of neurons in the motor cortex, so the frontal lobe, right? And the other body-shaped map of neurons back in the somatosensory cortex of the parietal lobe. So as you can see on that image we saw here before, the fingers, the toes, the mouth, some of those areas, they have tons of neurons dedicated to their function relative to the size of that body part. Whereas the trunk, even though the trunk is a big part of our body, right? There's not many neurons in the brain dedicated to processing touches in your trunk or to controlling muscles in your trunk. At any rate, studies like his are an example where we can sometimes use single cell recording in the human brain. And you can see how this might be more powerful than a standard brain scan that simply tells us, some chunk of the somatosensory cortex is active, but can't get down to individual neurons. Finally, I wanted to end by just mentioning some limitations of cognitive neuroscience, especially of brain imaging studies. So first, running a brain scanner is super expensive. It can cost hundreds of dollars to run one participant for less than an hour. So even with a big grant, this research can be hard to do, and the machine itself may cost millions. But that means we often end up with small sample sizes. We can't just keep adding participants, especially in some of the earlier studies when fMRI first showed up in the 90s. So for this reason, and for some other issues, findings from brain scans often get kind of overgeneralized or misinterpreted, or may even be false positives sometimes if we don't go back and replicate it in another lab. This was more from early on. So early on, false positives were a major issue because when you're measuring 1,000 or more tiny little volumes of brain area, you can't just run a simple statistical test on each separate area to see if it changed, since that's a buttload of statistical tests. Each one might have a little bit of a chance of being a false positive, so overall that hugely inflates your rate of false positives. Nowadays, thankfully, we do have better statistical procedures, but this problem is complicated enough that statisticians who specialize in brain imaging still argue about the details. Furthermore, brain imaging often provides only correlational evidence. Like, this area tends to light up during this task. And simply knowing that an area is active during a cognitive task, that doesn't really tell us how exactly it's involved in that task, the mechanisms by which it accomplishes that function. So we need to go further than just seeing what area has a little extra blood flow when solving a riddle or when imagining a smell or whatever. Also, if we're not careful, we can end up overemphasizing modularity, that old idea of localization of function. Even though nowadays, practicing scientists, those who do this stuff professionally, their emphasis is a lot more accurately on networks of brain regions, not individual brain areas, but networks of brain areas all connected together, and sort of the complex interplay and symphony of activity that arises from all sorts of feedback and bidirectional connections and input from all over the brain. That said, though, brain imaging has been freaking amazing. It's been an amazing technological breakthrough. It's taken us light years beyond where we were back when we only had animal models or studying people who happen to have a stroke or get an iron rod shot through their head. So in the big picture, we never use a single study in science to answer any scientific question definitively, right? You want to do replications, try different variations, stuff like that. Likewise, we never use a single technique to determine how something as complex as cognition happens in something as complex as the brain. So often cognitive neuroscience works best by combining various imaging techniques with non-imaging techniques and other evidence as well. Now we'll stop there for now, but I will have one last short video for cognitive neuroscience dealing with an important issue kind of anytime you interpret brain scan results, something called the reverse inference fallacy. We'll wrap up cognitive neuroscience with that, and then we're on to the next big topic.